Hello, everyone. Let's get started. Good evening. Tonight we have a two-part program. The first is the address of the president-elect, and then this will follow by the awards ceremony. I'm Vivian Negron-Ortiz, the president of the Botanical Society of America, and it has been a privilege and an honor to serve this year as president. Thank you. I am so pleased to be here tonight to introduce and to have a chance to be with our president-elect, Dr. Brenda Molano-Flores. She will be soon to be our new BSA president. A little bit, yes, a little bit about her. Well, her bachelor and her master are from the University of Puerto Rico, Rio Piedras, from the island of Puerto Rico. Her PhD is from the University of Iowa, and she is currently at the Illinois Natural History, where she holds a position as a research program leader. From rare plants, environmental DNA, species distribution modeling, surveys, demographic studies, species assessment, Brenda's research includes a wide range of ecological topics, all related to plant-insect interactions, reproductive and population biology, conservation, and restoration. Brenda is and has been a very productive scientist. Her research has been funded by more than 55 grants, has trained 24 postdocs and grad students, and mentored many, many other students. She's very well known within the BSA community, as she has served as chair of the BSA Ecology Session and the Community and Human Diversity, just to mention a few. Something that you don't know about is that we are both from Puerto Rico, but we didn't know each other until 2008. In 2008, Brenda participated as an invited speaker in a symposium that I co-presided in Vancouver, Canada. From that small interaction, we had conversations about rare plants and the importance of research in the Florida Panhandle. From there, Brenda, her collaborators, and her students, with grants from the state and the Fish and Wildlife Service and other sources, conducted a series of research and studies to advance the recovery of two threatened, endangered Florida species, all of them federal listed. Pinguicula ionantha, a carnivorous plant, and a mint, Malbraria alba. They did a remarkable job addressing scientific gaps, and those gaps and that information has been used for decision-making into the recovery of the species. And this is just one example, because she has worked with more than 15 different species. Brenda brings all these experiences, and I'm certain that she will serve with enthusiasm and fairness as BSA president. The title of her talk tonight is Finding the Joy, Tall Tales of a Plant Lover. Please join me in welcoming Dr. Brenda Molano-Flores.
Vivian, thank you so much for the introduction. And I believe that actually we started interacting even sooner than that. That's the thing. I mean, I remember sending emails to a bunch of agencies, staff from different agencies, and asking, hey, do you have money to work with rare plants? And believe it or not, Vivian was the only person that replied that email. So that is to show you that you have to put yourself out there sometimes, and that can lead to amazing things. So remember that. So today, I'm trying to remember this. Oh, yes. That's it. That's it. That's my talk. That's my talk. That's my talk. Before I start this talk, I want to thank a couple of folks. First of all, I want to thank all of you for electing me as your new president. Thank you for doing that. I don't know what I'm getting myself into, but I will do my best to represent all of you. And I would like to encourage all of you that to email me. And if you see something that you don't like, let me know. And if you like, please let me know as well. The other thing that I want to do is to thank the BSA staff. How many of you are here? I think that I see a few. Stand up, please. Stand up. Come on, come on. Stand up. And I want to also thank Melanie for putting this wonderful conference. Thank you. And I'm forcing the staff to stand up because the BSA staff, they do so many things behind the scenes. You know, stuff can be falling apart, and you wouldn't tell that they're falling apart because they are supporting us. So it is extremely important for me to thank you for all the help that you provide the president-elect, the board, because I know that it's not an easy job wrangling all of us. And sometimes, I mean, it is, to me, it's important that you know that you're highly appreciated. So thank you again. And I need to thank somebody that is very special, Marianne Feist. Marianne Feist is my wife. Marianne and I, we have been together for 24 years. We share a passion for plants. You can see that there. Most of our vacations are botanical oriented. It has been a joy. I mean, having a life with someone that appreciates plants. So thank you. And I have decided that because I am your Madam President, now around the house, I'm going to be saying, hey, you're the First Lady of Botany. So tonight, as I was thinking about the topic of this talk, when I put the abstract together, I started thinking, well, you know, we're going to be spending like 4 days going to workshops, reading a bunch of posters, listening to amazing talks and lectures. So I made the decision that I didn't want to give a data-heavy talk. Okay, we had that all week long. It was great. And also, I was thinking about the theme of tonight, which is celebrate, to celebrate that this conference has been wonderful to celebrate that next year we're going to be again in person, and that we're going to Michigan for Botany 2024. So I decided that today I'm going to share with all of you what brings me joy or has brought me joy when it comes to the botanical realm. And during this talk, especially at the end of the talk, I'm going to ask you what brings you joy within this botanical world. I want to hear from you about what makes you happy when we talk about plants. So keep that in mind, because I'm going to start pointing at people saying, what brings you joy? So as any journey, when it comes to discovering joy, this journey, my journey at least, and probably your journey too, has an origin story, like a superhero, like a plant superhero. And as Vivian mentioned, I was born in San Juan, Puerto Rico, Boricua. And I was born, actually, I grew up in the city. I was just like, a suburb of San Juan. And my comfort level was very small, maybe a mile away from my home, maybe 6 miles. That was it. I call myself the worst islander that you can meet because I don't like the beach. I don't like the sand. When it came to nature, even though that I was surrounded by beautiful tropical flora, it was just like, it's there. Also, growing up, there was this somewhat expectation, and I think that many of us feel that either our folks, they would like us to be doctors, lawyers, or accountants. And when I was doing my undergrad at UPR, it only took one professor to change my life. So to all of you that are teaching, keep doing what you're doing to encourage students to stay in botany, because that makes a big difference in my case. And I have heard that so many times during the meeting. However, I want to point out that my folks, even though that they were concerned, they actually got, my sister is a lawyer, my brother is an accountant, and I became a different kind of doctor, a plant lover doctor, but they were fine with that, and they were very happy with it. My mom was like, what can you do with studying plants? And I was just like, don't worry, Mom, I can make it. And because I wasn't 100% sure about this botanical world, I decided that I didn't want to go into doing a PhD right away. And I decided that I wanted to do a master, so then I could better understand, what I was doing. And during my master, I had 2 wonderful mentors, Charlotte Taylor, which is a Rubiaceae expert, and Jim Ackerman, an orchid expert. And both of the individuals, they were great mentors because they gave me academic freedom, and they were okay when I made the decision that I didn't want to do anything related to tropical ecology. So don't ask me about tropical ecology. But Charlotte Taylor was an amazing mentor. She was the first person that took me into a botanical expedition in the Dominican Republic. And that's when I realized, man, I can study plants and I can travel too. And we had awesome adventures, from getting stuck, and so it was amazing. once I finished my master there, then indeed I decided to pursue my PhD. And of all the places that an islander can go, I decided to go to Iowa. I don't regret it because in Iowa, I fell in love with the Midwest. I fell in love with the North American grasslands, which is a study system that a lot of my projects are within that ecosystem. And also, I had another great mentor, Steve Hendricks. He has done a lot of work with Pastinaca sativa, plant-animal interactions. But Steve was, again, another mentor because he didn't say you have to work on Pastinaca sativa. He pretty much said, okay, here's the key, go and develop a project. Go to the biological station and come back with a project. And that's how I end up working with plant breeding systems, habitat fragmentation, and prairies. And I appreciate that quite a bit. After that, I finished my degree. At that point in time, I decided not to do a postdoc. And then I ended up at the Illinois Natural History Survey with the University of Illinois. And there, I had another great early career mentor, Chris Whalen. Chris Whalen is not a botanist. He's an ornithologist. But in my case, having an early career mentor was crucial for me because he helped me meander some of the issues that some of you are going to learn very soon in terms of human resources, grants and contracts. So he helped me meander that landscape. But one of the best things about working at the Illinois Natural History Survey is that I work with an awesome group of people that they love plants. They are always engaged. They're always happy, even if it's 100 degrees out there, even if there's tons of ticks, mosquitoes. Has been a place where I have been mentored by these individuals. I have been able to mentor them and develop this very close relationship of collaboration. And also because I'm within an academic institution, I have been privileged to mentor students. And this brings a lot of joy. For once, I'm learning from my students, and I'm able to tell them, sometimes things are going to work out, sometimes they're not going to work out, and that's fine. That's fine. And I have been privileged to mentor undergrads, grad students, postdocs. And one thing that I want to point out is that also during my late career, I have had a very special mentor. I always say that it's never too late to be mentored. And one person that has provided a lot of mentoring during my late career is Dr. Janice Kuhn, emeritus professor from Eastern Illinois. And I believe that Janice is here. Don't hide. And Janice has been an instrumental person in terms of collaboration, mentoring, being honest with me. Even in my late career, sometimes I want to do projects that I keep adding. And she is the one that will tell me, no, Brenda, we have to go back to the proposal and do what we proposed first to be able to add anything else. And one of the best things about Janice is that she knows my vices. She knows that I need coffee and blue M&Ms. So it is wonderful to have somebody that knows what I need to be functional. And when you work with somebody that is always smiling, no matter how difficult I can be, that is a plus as well. So all of this is to tell you about the joy of mentoring and how important mentoring is. And the things that we do as advisors, the things that we're doing as part of this society. BSA, we're putting a lot of effort into mentoring, and that's one thing that I appreciate and that we want to keep doing. Because a mentor can either turn you on or turn you off when it comes to enjoying plants. So I have a few points, pieces of advice. One, for all of you, seek mentors as early as possible and continue throughout your entire career. Seek mentors that care beyond the research, that care about your career goals, that, if something is going on with you, that they will ask you, what's going on? And also seek mentors that are honest with you. The one thing that I love about Janice is that she will tell me when I'm wrong. She will tell me, no, I don't think that we can do that. And that's extremely valuable because sometimes, you can be hitting a wall and you don't see it. And for people that are in a position of mentoring, be the best mentor that you can. You're in a unique position to change someone's life. And I will say, don't be like this mentor. So please, yes, do not pretend and stress your student. I don't know if that is legal or not, having the cartoon there, but hey. Another place that I find a lot of joy is through the process of discovery, through the process of doing the research that we do. And I consider myself a plant ecologist. I do occasionally ecosystem level research, community level research. But the part that I enjoyed the most is to do single species research, do species at the species level, because that's the place where I can learn a lot about the biology and ecology of a lot of the species. And I put this collage here because this is just a few of the species that I have studied. For each of these species, I have learned a lot. Like for example, we did a project with Lobelia spicata. I learned a lot about gynodioeces, having female plants, hermaphroditic plants, and how population size can affect the ratios of the species and how pollinators can be affected. I did a project with Eryngium yuccifolium, a prairie species, where if you have florivory, that's going to affect pollinator visitation. I did another project with Tradescantia ohioensis, where we discovered that if invasive species are present within the environment of this species, fruit set and seed set is going to go down. So each of these different projects have taught me something cool, something new. One project that I have enjoyed spending a little bit, some extra time, is a project that I have with carnivorous plants. And when I tell people that I work with carnivorous plants, they immediately say, oh, the Venus flytrap. I say, no. Is it the pitcher plant? No. Is it the Drosera? No. I work with pinguiculas. And pinguiculas are an awesome group of species. They're found in the United States, in Central America, Europe. There's more than 130 species of pinguicula that have been described, and they continue to describe this species. And in my case, I have been privileged to work with 3 pinguicula species: Ionantha, lutea, and planifolia. And these carnivorous species are very cool. I mean, they have flowers with different petal colors. The leaves, they look different. And one of the coolest things about working with this species is that we work in the Florida Panhandle, an amazing place to do research. We stay in a biological station where you have this beautiful scenery, gray sunsets, and that music, you know, goes with the, the scenery. So it is an amazing place to do research. We enjoy, you know, riding the ATVs and to go and collect our plants. And I'm always proud that I give the best advice to my students when we're working there. I can tell them, yes, we can drive through that model, you know, a pot of, you know, and discover that we cannot do it. And then humbling, we have to walk a mile to be able to get reception, so then somebody can get us out. So maybe do not listen to your advisor, you know, when you see something like that. But working with Pinguicula species has been amazing, partially because when we started the work that we have done, we realized that when it comes to North American pinguiculas, hardly anything was known. I mean, we knew, you know, the locality, but we didn't know hardly anything about the biology and ecology of the species. And because of our collaboration, Vivian, we were able to learn, and we keep learning about this amazing group of Pinguicula species. I'm going to tell you a few things that we have learned. One thing that we didn't know anything about was about the breeding system of the species and of these three species. So we spent 2 years studying the breeding system, putting organza back, doing hand pollinations, and we were able to determine that these species are self-compatible and that they have a high fruit and seed set. We also learned more about all the different types of pollinators that are visiting this Pinguicula species. And something that was pretty cool is that bumblebees provide a lot of ecosystem services when it comes to this Pinguicula species. And they do the classic bus pollination. And it's just like they come, boom, they're gone. We learned that. And also occasionally, we will see a few honeybees visiting this Pinguicula species. The other thing that was very cool that we learned, and nobody knew about this, is that actually the flowers can track the sunlight, the sun. So we put some cameras, and I'm going to advance, but pretty much is that for most of the night, the flowers are going to be drooping down. By midday, they're already up. And then by later during the afternoon, they're going to start dropping back down. And then the process starts all over again the following day. And again, nobody knew about this. Another thing is that if you're looking for a study system to study floral longevity, Pinguicula species might be an awesome system to work with. We did a project where we did hand pollinations. And if you pollinate the stigmas, it just takes a few days for the corolla to fall to the his. But if the flowers are not pollinated, they might be around for 2 weeks, 3 weeks. So this could be an awesome system to study floral longevity. I don't know why. I mean, they need to be pollinated, but I mean, the energy cost of having such a big inflorescence, why? So grad students, think about projects here. And another thing that was very, very neat is that although we knew something about the prey that the species were capturing, for example, springtails members of the Diptera family. Again, we didn't know anything specific about the community, the prey community. So we studied that. But what I think that it was the coolest thing that we did when it comes to prey capture is that we developed a very simple, technique where we use white electrical tape and we just peel the insects from the leaves. Very cost effective. It doesn't damage the leaves. And that was important because a lot of the species are listed either at the federal level or at the state level. So we wanted to have a technique that was not going to damage the plant. And as a result of all these different projects, we have generated publications that bring some level of joy. But what brings the most joy for me is that during this entire process of discovery, I have been able to involve students, postdocs, collaborators, and even community scientists as part of this entire process. And to me, that has been an awesome experience. And because, if you are doing research projects, you need funding to do those projects as well. And when we think about funding, many of us, we go, oh, NSF is the only place where you can get funding to do basic or occasionally applied research. And many of us here, we have been successful about getting NSF. Some of us, we have had maybe 1 or 2 NSF grants. There is a period of drought there. And sometimes, we don't get it. Okay, that's the reality. However, I want to point out that this is very, very stressful, especially for early career scientists, for postdocs, for students in general. And what I want you to tell you is that you should embrace the joy of diversifying your funding portfolio. There are many agencies out there that they have a lot of funding that will support a basic applied research program. The Department of Defense, the USDA, the DNRs of your state, foundations, corporations. There are a pot of money out there that we should consider. Because if you do that, then I can reassure you that your life will be less stressful, that you might be able to go take a bike ride without thinking, oh, I need to submit a proposal. You might be able to go dancing, and I hope that we're going to be doing that tonight without thinking, or karaoke. Hey, maybe you can karaoke that I don't need. Anyway, funding. The other thing that is extremely important about this joy of diversifying your portfolio is that if you work with federal agencies, the staff of a lot of these agencies, they want to go out with you and help you. They're eager to do that. And they might help you to count plants. They might help you set up a prescribed burn study. They want to help us in general. And if they don't have the money, they're willing to give you in-kind contributions. They might be willing to give you an ATV or 2, a boat, even a helicopter. So that is amazing. But I have to admit that the projects that I enjoy the most are the projects that are on a budget. Those projects that either you don't have any money, and you want to do the research, or just maybe 1,000 here and 1,000 over there, because they made me be as creative as possible. So if I need to drive from point A to point B, I need to get a car. And if I don't have too much money, I cannot be picky about it. So I always try to get the best deal that I can when I'm doing my field work. Also, when you're doing field work, we might not be able to buy airline tickets. So we have to do road trip from point A to point B, family road trip. So why not take the moment and stop at special places? Why not, if you go to Minnesota, stop by Austin, Minnesota, and go to the Spam Museum and take a photo with Spammy. You can do that. Or if you travel to Alabama and you go through the town of Dothan, Alabama, you will learn that Dothan, Alabama is the peanut capital of the United States. I learned that. That's an awesome factoid for trivia night. And you can always stop and take amazing photos of roadside art. I love this metallic bull. The other thing is that the accommodations can be extremely fancy as well. And believe it or not, an office space can become a hotel room. With the exception that by 7 o'clock you have to be out because the staff is going to come to start working. So that's the only downside about that. And the other thing is that I'm always trying to find creative ways to use the equipment that I have. I don't need a lot of money, but it costs money. So when I'm doing field work, I'm looking at my organza bags. How many of you use organza bags? Yeah, you use them, to pollinate, to exclude pollinators. So I'm always thinking, okay, what else can I do with my organza bag? What other creative thing I can do? Well, I can tell you that I can make a mean cup of coffee with that organza bag. So if you're in a pinch, you can use your organza bag. So even though, that all of this, is trying to find creative ways to do your research, sometimes, you bump into issues. And, you have to deal with those issues. And recently, I have been involved with a project that I figuratively and relatively hit the wall. And that brings me some sadness because of geopolitical issues. It brings me sadness because habitat is being destroyed because of socioeconomic issues. However, I want to point out that during those circumstances, I try to talk to people. And in this particular case, this is very close to the Mexico border. The habitat of the thorn scrub portions is being destroyed, and multiple listed species are found within that habitat, such as the Zapata bladder pod. And although that makes me very sad in terms of what is happening, I always do my best to find common ground with the landowners, with the people that are doing surveillance or the members of different agencies, and talk to them and try to find that common ground that, that habitat is important. There are plants there. Let's work together to do our best to protect the habitat under the circumstances that things are happening. So that's one thing that brings me joy in terms of connecting with people and talking to them about multiple issues when I do the work that I do. The other thing that I'm going to point out is that I also like to embrace the joy of keep learning. I think that that's extremely important. Like, it doesn't matter if you are starting, if you're mid-career, that joy of I need to keep learning. There's so much out there that I need to know. To give you an example of that, I do a lot of plant-animal interactions. I focus on the plants. I'm very happy that if there are, bees, bumblebees, butterflies, flies, I'm okay with that. But I have realized that I need to kind of like dive deeper and identify all these different types of pollinators. So recently, I decided to take a bee course. I went to Maine. I spent a whole week there learning about native bees. I spent time with an amazing group of people. I discovered this suction gun that you can go and trap insects, and that was awesome. But the best part was that last week, I'm walking around my neighborhood in Madison, Wisconsin. And as I'm walking, the neighbors sometimes wonder about me because I'm just like, oh, she's looking at a tree. She's like standing like that. And so my next door neighbor, I'm walking, and I see this bumblebee. And I'm just like, no, it cannot be. I went back home, got my phone. I started taking all these photos. And it turned out to be that it was the rusty patch bumblebee. And it was just crazy. Yeah, I mean, it's like, whoa, it's the rusty patch bumblebee. This is the most endangered insect that we have in the United States. It was listed, I believe, in 2019. And I was privileged to be able to see that bumblebee around my neighborhood. And then I was just like knocking on the doors and telling my neighbors, here, you have the rusty patch bumblebee, and thanking them for planting native plants. And they're like, oh, okay. But the other thing that I'm having a blast is embracing new technology. As we get old, learning R. So I'm trying to embrace that process. So one field that I've been collaborating with new young scientists is the field of environmental DNA. And environmental DNA is becoming a very popular field. And when we talk about environmental DNA, we're talking about we all shed cells. They're in the environment. You can find them in the aquatic system, the air, terrestrial system. So I've been collaborating with an amazing group of people. And actually, during Botany 2020, we put together a symposium to tell our botanical community about this new emerging technique to sample biodiversity. And one of the things that I have been highly involved is to help with projects where we are developing protocols in terms of how to collect specimens, how to collect material, so then we can extract the DNA from flowers, for example. And now we're getting into asking ecological questions about plant-animal interactions using environmental DNA. So one of the first projects that I was involved with, I was the one that was in charge of developing this amazing flower community in the greenhouse with different floral resources, pollen, nectar, different flower shapes. And then I purchased a bumblebee nest. And then we released the bumblebees in this secure environment. And then we went and collected the flowers and tried to assess, are we detecting bumblebee DNA? We asked the question, if you have a flower that has tubular shape, is that going to make a difference? If it is an aster flower, is that going to make a difference? Because we want to make sure that we are providing information and making sure that environmental DNA is an effective tool to do bio-monitoring. Now, because we chose to diversify our funding portfolio, we got some funding from the Department of Defense. And the DoD has a lot of land that they do a lot of training, military training. And the DoD is one of the largest land holders of areas that have a lot of T&E species. So they have to protect what they have, but also they need to do the training too. So it's a difficult balance that they have, that they're charged with. So we receive funding to be able to assess if environmental DNA can be an effective tool to determine can we find rare pollinators within temperate grasslands, tropical rainforests, and desert environments. And this is important to test this particular tool in as many environments as you can, because it might work in a grassland, but it might not work in the desert. So we need to sort out all those different things. So for example, in Wisconsin, we are working to develop protocols that are going to assure that we're capturing the entire community of pollinators associated with a plant or associated with an entire community. So we want to know, okay, how many flowers do you need to collect to be able to reach that plateau of maximum pollinators or flower visitors? We're also trying to make sure that environmental DNA as a sampling tool is a cost-effective tool. So we have done a project where we have people observing plants, writing down the pollinators. We have put cameras, and then we have gone and collect the flowers and compare if indeed environmental DNA is as effective as having a person in front of a plant, or if you just put cameras there. The other thing that we have been doing is that as part of this project, we were able to go to Hawaii, which was an amazing experience. And part of the reason that we're going there is because we want to, again, compare the different types of pollinators that are visiting native plants, non-native plants, and rare plants. And I have to admit that going to Hawaii was awesome. I was able to take a helicopter ride. I felt like Tom Cruise in Top Gun, suit up and everything. And the coolest thing was that I was able to see some of the most pristine forests in Hawaii all the way to the top. And I was able to collect specimens within this amazing forest. And also, I was honored to work with an amazing group of plant conservation biologists in Hawaii. And it was a very humbling experience because they have a tough job in Hawaii. And not only that, it was humbling because I learned about the cultural value of this plant, the cultural value of some of this pristine forest that they have. And it was an amazing experience exchanging that knowledge. And also, I was very humble that I was in the presence of some of the most rare plants that you can find in the world. I mean, going to some of these sites and seeing maybe just the last plant or the last populations, it was a reality check for me. And also to learn about all the efforts that they're doing to make sure that at least we don't lose more of this species. Lastly, I want to point out that this journey into studying environmental DNA has taken me to Arizona. Right now, our team is studying saguaros, saguaro flowers. And doing this work when it's like 110, it's not fun, I can tell you that. But we have a project where we are collecting saguaro flowers within this urban to rural gradient, because we want to know if there are differences in the type of floral visitors that are going to these flowers. So all of that has been an amazing experience. So I want to leave you with a few thoughts. When it comes to finding the joy of being mentor and mentoring, the joy of collaboration, the joy of discovery, the joy of creativity, the joy of always being open to trying and learning new things. And also, why not? The joy of being silly. And with that, I'm going to ask you now, what brings you joy within this botanical realm? Thank you, and enjoy the rest of the meeting. What time is it?
We have about 8 minutes.
Oh,
We have time for questions.
actually, I just want to learn about your joy. Who's the first one? There you go.
As some of you know, thank you so much. I love alpine plants, and I have the joy of this little itty bitty tiny flowers. It just makes me so happy, like right there. Little itty bitty.
No.
Hello. Ooh. So for some of my species that I study echinacea, and so I work in the cedar glade habitats. And so I get the joy of looking at my nice bright pink flowers.
Thank you.
Hi, I'm Brian. I study agave, and I find joy in, my international collaboration and meeting new people and learning about new cultures.
Thank you.
I find joy in learning about other people's stories of plants and the way that they relate to them.
I need to find joy around here, too. Come on.
I find joy in learning about the plants that are in my neighborhood, in my city, in my state, just learning more about what's around me.
I'm Ryan. I work on polyploidy and rhododendron, and even though that's frustrating, it does bring me a lot of joy. And my first Botany Conference was actually in Boise in 2014. I came with my lab from Colorado, where I was at the time, and I have to say that this conference brings me a lot of joy. I love coming back to this, and they've been supportive since I gave my first poster, my first talk, and so thank you. Brings me a lot of joy.
Thank you.
I find joy in seeing hawk moths visit flowers because they're just so cute.
Hi, I'm a paleobotanist, and I find joy in the fossil plants, even though they're dead. But I find joy in camping with the people you love, making new friends, and just being together in camp with camaraderie, like you said. It makes me love it even more.
Awesome.
I find joy in plants that are growing in unexpected places, places that look inhospitable. I love finding the plants that are growing in the brick walls and the sidewalk cracks, the resilience of plants.
Hi, I find joy in the smell of an herbarium cabinet when it hasn't been opened in a very long time.
I am also a paleobotanist, so I find a lot of joy in that we can shine a lot of light on what objectively feels like it should be impossible to learn about, like 300 million years ago, where I'm studying.
Oh, okay. There's a lot of joy. I feel a lot of love here.
I find joy in cooking for people and listing out how many plants that they just ate.
I find joy in watching students learn and grow and learning about more plants than I know.
Awesome. Over here.
I find spending 10 days with my daughter out in the field and at the meeting. And I also find the inspiration with the young people that are collapsing on the ground and looking at plants and just they're so enthusiastic. It took us 4 hours to go 3 miles. So.
One more.
I study development and structure, and I find joy when plants don't follow the rules that we were taught in class.
One over there. Well, you ask and you receive.
I find joy in soil.
Woo!
I find joy in my bad-ass students who are constantly pushing new frontiers. It's really cool.
So I'm going to say thank you so much for coming. To me, having all of you here is an amazing experience. And as I mentioned before, I will do my best to represent you, to hear you. And again, I want to thank you. It brings me joy. So.
All right, so now welcome to the annual awards presentation of the Botanical Society of America. But I have to say, I find joy having you here. And because I work with rare plants, I find joy just finding them. Okay, we're going to honor tonight and recognize several of our BSA members. I would like to recognize and thank the Society Partners Presidents for another fantastic meeting. Could you please stand up when I call your name if you are here? Scott Chudy and Daphne Stone, American Biological and Lichenological Society. Stand up, please, if you're here. Okay. George Durr, American Fern Society. Josh, hey. Eric Rolson, American Society of Plant Taxonomists. No. And I know that Patrick Herendon is the International Association for Plant Taxonomy is in Brazil. And Anna Monfils, Society of Herbarium Curator is not here too. But please thank them. Special thanks is for our overall conference program director, Dr. Melanie Link Perez. for helping us to put our scientific program here in Boise. Thank you, Melanie. I also want to recognize our field trip coordinator, Jim Smith from Boise State University. If you're here, please stand up. This is what I heard. I heard it was hot, but I also heard that they had a great time exploring. We also owe a very special thanks to Joanne Stogrand, our conference director. Please, Joanne, are you here? She's busy. Okay. And her dedicated support team, Kevin Stogrand, Steve and Catherine Bornhoff, Jessica Stogrand, and Diane Killian for the logistical support for all the attendees and sessions during yet another fantastic botany meeting. Thank you. Thanks also to all the BSA staff. Katrina Adams, Rob Brandt, Heather Kennedy, Jennifer Hartland, Richard Hent, Tricia Jackson, Amy McPherson, Amelia Neely, and Beth Parada for outstanding support to all of us. Please stand up. Thank you. And now we have 3 special awards. To our outgoing board members for their service, contribution, and leadership. Please come and join me. Michael Donahue, past president. Chelsea Spetch, Director at Large for Diversity, Equity, and Inclusion. Chelsea? Please join me. Hey, Chelsea. Our student representative, Ioana Angel, is not here, but we will send her, but let's congratulate her. As you can see, I have a script, so that's what I'm reading. Now, for your information, the BSA provided about $140,000 in award and travel support this year. First, we have some very special student research award that we would like to recognize. The Society gave out 32 Graduate Student Research Award and 1 Carlin Research Award, which provided $1,500 to each of these students. They are rising stars in our field, and we are extremely proud of their work. I would like to acknowledge Jordan Argret from the University of Georgia, winner of the Carlin Award. Please stand if you're here. No here. Okay. The research proposal placed first among their peers. So now, will all those receive a Graduate Student Research Award please stand if you're here? No? Just one. Yeah. Thank you. We also provide research awards of $500 to undergraduate students researchers, and this year we provided 6 of them. You will see those winners in our slide presentation. BSA provides 2 significant dissertation research awards. First, the Donald R. Kaplan Award in Comparative Morphology. This $10,000 award was created to promote research in comparative plant morphology and support the PhD research of graduate students in this area. This year's winner is Haley Nedblake. Yes. University of Kansas for the proposal evolution of B-exclusionary corolla with differences in penstemon. Congratulations. Second, the Graduate Student Dissertation Award in Phylogenetic Comparative Plant Biology. This new 10,000 endowed award is courtesy of a longtime BSA member and anonymous donor. It was created to promote research in comparative plant biology and support the PhD research of grad students in this area. This year's winner is Zachary Moskowitz, University. University of Connecticut for the proposal, The Evolutionary Dynamics of Lichen Symbionts, Going Global. Congratulations. Are you here? No. Okay, there are numbers of travel awards that were also given out, including the Shield Award, the Plants Award for undergraduates, student and postdoc travel awards, Developing Nations Travel Awards, as well as various travel awards from the sections. If you are a student or educator that won any of our BSA or sectional travel awards, will you please stand and be recognized? In addition, will all those receive the Best Paper or Best Poster award please stand up? Yes. Now, these people work very hard and are being recognized for their excellent job with their talk or poster here at the conference. Congratulations to you. We also wish to recognize undergraduates who were nominated for and received the Young Botanist of the Year award. These are undergraduates who have passions for plants and will become future leaders in our field. We hope that all of these early career scientists will continue their affiliation with the BSA and join us next year in Michigan and at Botany Conference for years to come. The Society also supports public policy efforts in science by providing 2 policy awards that send early career scientists to Washington, D.C. for training and meetings with representatives. The 2 winners for this year's Public Policy Awards are Catherine Charton, University of Wisconsin-Madison, and Zach Quirk, University of Washington. Are you here? Yes. Congratulations. We have another award in the area of public policy called the Botanical Advocacy Leadership Award. This award is organized by the Environmental and Public Policy Committees of BSA and ASPT, support local efforts that contribute to shaping public policy on issues relevant to plant sciences. The winner this year is recognized for the proposal called Set a Day at City Council, Social Actions for Integrative Botanical Education, Access to Land, and Food Sovereignty. Congratulations to Maya Shamshid, Dean. Now, related to publications, the Society provides the Grady L. and Barbara D. Webster Structural Botany Publication Award. The American Society of Plant Taxonomy and the Botanical Society of America are pleased to join together in honoring both Grady and Barbara Webster, longtime members of our societies. In odd years, the BSA gives out this award, and in even years, the award is provided by the ESPT. The Webster Award recognizes the most outstanding paper in the American Journal of Botany in the field of structural and development botany. This year goes to Mark Olson and co-author for their paper, The Vessel Wall Thickness, Vessel Diameter Relationships Across Woody Angiosperm. Congratulations.